This is a production of Cornell University.
Thank you, Susan, for the introduction. And it's always nice to talk to your colleagues about your work. So today I'm going to tell some of our recent research on crop nutritional quality improvement. So my lab has been focused on mainly 4 areas. One is the carotenoid research, and the anthocyanin, selenium, and the micronutrient biofortification. And today I'm just going to tell you some of our recent research results on carotenoid research. So this is a slide I always like to use to introduce carotenoids. So carotenoids are not new to any of us because carotenoid gives the red, orange, and yellow color found in many vegetables, fruits, and flowers. So why are we interested with carotenoid research? This is because carotenoids are known to be important in human nutrition and health, like in reduce the risk of cancer, heart disease, and age-related eye diseases. And it's some of the carotenoid we called provitamin A carotenoid, like a beta carotene contain 1 or 2 beta rings, and they are known to provide the precursor for vitamin A synthesis. Since our humans can't synthesize the vitamin A, so all of the vitamin A in our diet can be ultimately traced back to carotenoid origin. And many staple crops are known to contain very low level of carotenoids. As a result, they contribute to the global prevalence of vitamin A deficiency. And as you can see, vitamin A deficiency affects a large number of preschool children and pregnant women in those poor areas. So typically, carotenoids are C40 terpenoid compounds. The carotenoid biosynthetic pathway has been very well established over half a century ago. And the many carotenoid, because they double bond and ring structure, give distinctive colors. Like we know tomato is red color because tomato accumulate a lot of lycopene. And carrots is orange color because accumulate lots of alpha carotene and beta carotene. Many yellow flower like a daffodil is yellow color because accumulate lutein and other xanthophylls. So during the past 2 decades, we also start to understand the process to produce apocarotenoids include some plant hormones, ADA, and stracholactin, and also beta ions. They are volatile compounds to give flower fruits the flavor and aroma. So all the genes in the carotenoid biosynthetic pathway have been major gene. All the genes in the major carotenoid biosynthetic pathway have been cloned and well studied from many crops and plant species. However, till now we still have a very limited knowledge about the transcription factors, and the Richter genes control the expression of those pathway genes. So carotenoids essentially are synthesized in plastic in plant tissues. So carotenoid can synthesize in nearly all kinds of plastic, like amyloplast, chromoplast, and chloroplast, etc. But they accumulate in high levels in chloroplast in green leaf tissues, and also in chromoplast found in many flower, fruits, and vegetables. Since the ultimate goal of our research is to understand how plants can synthesize a high level of carotenoids, so this is our focus, to try to understand how carotenoids are synthesized and regulate in chloroplast. So for many of you know, for a long time, my lab has been working with the orange gene that was originally cloned from orange cauliflower. and use the map-based cloning strategy. We cloned this gene and found it does not encode an enzyme involved with carotenoid biosynthetic pathway, but instead it encodes a protein with one unload function and contain DNA-J cysteine-rich domain, but it's not chaperone protein by itself because it's lack of the J domains. So recently, in collaboration with Israeli scientists, we found that the OR gene is also controls beta carotene synthesis in melon fruits. So our collaborator from Israel, and they made a cross between the orange melon fruits Delcy with the orange green melon fruits Tamijul, and in the F2 population, in this particular population, they found the orange fruit flash segregates with 3:1 ratio. Essentially, this trait segregates as a monogenetic dominant trace. So they did association analysis, and they found that the OR gene completely co-segregates with the flash color in this F2 mapping population, as you can see, with green flash fruits contain this band and orange contain this band. So in addition, they also surveyed the wide genetic collection they have, and they found the OR gene existed in 2 haplotypes. One is associated with green flash from all of those germplasm collection, and one is associated with either yellow or either orange or green or white. So in collaboration with them, we clone the OR gene from green flash and orange flash fruits, and we transform them into plants. As you can see, the gene from orange, the OR gene from orange flash fruits can induce carotenoid accumulation like the orange cauliflower gene. However, the gene from the green flash fruit can't introduce additional carotenoid accumulation, just like a wild-type control. So we looked at the cDNA sequence between these 2 varieties. There are 6 SNPs, and 5 of them cause silence mutation, but 1 of them cause missense mutation. So essentially, the later genetic variation show that in green flash fruits contain the G, and in the orange contains the A, and with the change, it's called 1 single amino acid change to change the arginine in the OR into histidine in the orange OR. So to see if this SNP or this single nucleotide difference is indeed responsible for the fruit flash color, and the nucleotide was mutagenized. So what we did is that we changed the G into A in the green gene, and we changed the A into G in the orange gene. And as you can see, the green gene now can induce carotenoid accumulation, and the red genes can't introduce carotenoid accumulation. So this will confirm the effect indeed this SNP is responsible for the OR function. And we called this SNP golden SNP. And we call it golden SNP because with this single SNP change 1 amino acid, and it can cause beta carotene accumulation and also change the fruit color. And this golden SNP is responsible for all the mellow color difference, at least from those germplasm our collaborator collect from all around the world. So the next question we asked was, does this golden slab also work in other plant OR gene? So what we did is that we changed the conserved, we did site-directed mutagenase to change the conserved arginine. In this case, in Arabidopsis OR gene, changed arginine to histidine, phenylalanine, lysine, and alanine. And we also changed the nearby arginine into histidine and transformed those constructs into plants to look at the color change. And as you can see here, when we alter the conserved arginine into histidine in the Arabidopsis OR gene, and it causes the carotenoid, it causes the color change in Arabidopsis cali. And when we looked at the color change and found that the color is indeed associated with carotenoid, increased carotenoid accumulation. And more importantly, if we look at the profile, and we found that the beta carotene ratio in the white colors only 15%, but this become over 50% during 2 weeks of dark incubations. And in addition, we found the histidine substitution in a crop OR, in this case sorghum OR gene, it also causes the same effect, lead to high level of carotenoid accumulation. Moreover, we found that the histidine is not the only amino acid change showed to be effective. We showed that the alanine replacement in the OR gene is also equally effective to promote carotenoid overproduction. As you can see, the color culture becomes dark orange and showed a high level of carotenoid accumulation. So this experiment shows that both OR his or OR alanine can serve as effective genetic tools for carotenoid manipulation. So what does this finding mean for carotenoid improvement? And sometimes when we first clone the cauliflower OR gene and we tested its effect to improve crop nutritional quality, and in collaborate with Joyce Van Eyck, and we put the cauliflower OR gene into potato tubers, and the OR transgene does not only induce carotenoid increase in the fresh tuber, but during the storage, it stimulates continuous accumulation of beta carotene. As you can see here, following 5 months of storage, the carotenoid increased over 10 times in some lines. And furthermore, in collaborate with Mike Mizor, we also look at the carotenoid accumulation during storage of the squash. And as you can see, the color becomes darker during storage, and it's also associated with increased carotenoids. So what this means, the significance of this finding is that we know staple crops with, as I just mentioned, staple crops with low level of beta carotene contribute to the global prevalence of vitamin A deficiency. And we also know that the beta carotene in staple crops is not very stable during storage, because in most of the cases, those beta carotene are synthesized and stored in amyloplasts. So through this 2 OR transgene experiment in potato tuber and also the squash experiment, we know that the OR gene, or in the squash case, both cases, they provide some unique mechanism to improve beta carotene synthesis and also stable storage during the and beta carotene stability during the storage. So if we can find the mechanism, maybe we have a way to improve our staple crop with enhanced beta carotene level. not only for continuous synthesis, but also stably storage during post-harvest storage. So I showed you that in orange cauliflower, we know the mutation is due to an insertion of large transposon elements. However, in the melon case, the mutation is due to a later variation with the single nucleotide difference. So because this is single nucleotide, we prove that the single nucleotide change a specific amino acid, and it change the functionality of the OR to promote carotenoid accumulation. So many of us know that genome editing becomes the technology to use to change our genome to enhance, to improve agronomic traits. And so we are waiting for the maturity or advance of the technology so we can specifically replace one or two nucleotides to change one amino acid to see its effect in improve beta-carotene accumulation in staple crops. And actually, in collaborate with Peter Baer, the inventor of golden rice, they are trying to put our OR-His. They are trying to manipulate the rice OR gene to change the histidine, to change the arginine into histidine, and see if it can help, indeed can help improve the total carotenoid level in golden rice, and most importantly, see if it can help stabilize the beta-carotene during post-harvest storage. And also we know that, So we know that there's tilling methods and also looking for later genetic variations. So through those approach, we are hoping in future we can improve the staple crop for beta-carotene level, not only through transgenic approach of we know it's not in good stage to do that, but we believe we can use some way to improve, to create the functional allele of the OR gene in staple crops through traditional breeding approach. So maybe some of you already noticed that in our experiment we expressed both wild type OR gene and the mutant OR gene, and although both of them showed a similar level of gene expression and also similar level of protein expression, but they showed dramatic difference in induced carotenoid accumulation, as you can see both the color and the total carotenoid accumulation. So one question we were asking was, how does the OR-His or aniline or the OR mutant differentially regulate carotenoid accumulation? So to answer this question, we try to ask what is the function of the Y-type OR gene. So to answer this question, we were looking for OR interacting proteins. To find the OR interacting proteins, we use the co-immunoprecipitation in conjunction with mass spectrum analysis. So essentially, we have Arabidopsis plants express the OR Y-type gene tagged with GFP and the GFP only. We extract the protein from these plants and mix with beads, and then elute out the co-immunoprecipitated protein. And in collaboration with Ted Tannhauser at our USDA unit, and his lab analyzed those gel band and identified around 19 proteins that can be, this is 19 conserved protein. Actually, from each gel length, we identified more than 100 proteins. So we essentially, from multi-biological replicate samples, we identified 19 proteins that as potential candidates for OR interaction proteins. Very excitingly, we found PSY or phyton synthesis as one of them. So what is PSY? The PSY or phyton synthesis is the rate-limiting enzyme in carotenoid biosynthesis pathway, and it's also the first enzyme in this specific pathway. There are lots of experiments to show that manipulation of the PSY expression can have a profound effect on total carotenoid accumulation. And here I show you some example, like a golden rice. Here's the golden rice 1 and golden rice 2. You can see the carotenoid level is quite different. Actually, the only difference between golden rice 2 and golden rice 1 is change the PSY. And golden rice 2 use high active PSY from maize, and golden rice 1 use PSY from daffodil flowers. And also here's the golden canola. It's just express the PSY. You can see the canola seeds color change from green to orange color, and there are numerous experiments showed you express any PSY from any plants. Normally you can get orange colors showed like here. So to confirm that the PSY is indeed the OR interacting proteins, we use the 3 different approach to confirm that. And first through yeast 2 hype assay, and as you can see, not only cauliflower OR but Arabidopsis OR, there's a larger OR-like proteins. They all interact with PSY in yeast 2 hype assay. And we also did a pull-down assay to confirm such interaction. As you can see here, when you mix the PSY tagged protein with OR tagged protein, you can see the OR pull-down with GFP, and you can detect with CMAC, showed the direct interaction, but not in the selective controls. And the third approach, we used BIFC and further confirm such interaction in vivo in plastic. This is the site of carotenoid synthesis. So what does this interaction mean, and how does this interaction affect the PSY expression? So we generate the OR overexpressed line. We also got the lockout, and also generate the double lockout. As you can see with those Arabidopsis lines, they showed dramatic difference in OR protein and gene expression. And then we looked at PSY gene expression. As you can see here, if alteration of the OR expression does not affect gene expression, then we looked at the protein level. And as this slides show you, with the overexpress of the OR genes cause increased accumulation of PSY. And in the single lockout, the PSY protein level is very similar. We think they complement with each other. But in the double lockout, the PSY protein level was significantly reduced. And this is showed in leaf case, and similar in roots, you see the similar pattern of the change. So how does OR affect PSY abundance? Now we know with overexpressed PSY, reduced PSY, overexpressed OR, reduced expressed OR, and it causes PSY change. So how does OR affect the PSY abundance? So what we did is we introduced the PSY GFP tagged protein into Y type or Arabidopsis, and also into OR background Arabidopsis plants. And then we extract the protein and inhibit with CHX, the protein synthesis inhibitor, and looking for the PSY GFP protein level. And as you can see in the OR background, the turnover rate of the PSY was significantly, dramatically reduced compared without the protection of OR. So in addition, our collaborator Ralph Welsh from Germany, and he also looked to see if they increased the PSY protein level, is enzyme active or not. Quite often when you upregulate a protein, it can be just the protein level upregulate, but not necessarily means active enzyme level. So from his result clearly showed that with overexpressed OR, increased the PSY protein level cause increased enzyme activity. As you know, that PSY is the key enzyme in the carotenoid biosynthesis, and then the correlate accumulation of carotenoid was observed in leaf tissues when we have overexpressed OR, and when it's significantly reduced when the OR level was reduced. So in summary of this part, essentially we showed that both OR and OR-like directly interact with the PSY to enhance the PSY protein level and activity through regulated PSY protein stability. And because PSY is the key enzyme in the carotenoid biosynthesis pathway, it causes increased carotenoid accumulation. And actually, this is one of the first case demonstrate a protein can act as the post-transcriptional regulator of PSY to regulate carotenoid accumulation. So let's come back to the same question we tried to answer. So how does OR mutant differentially regulate carotenoid accumulation in both of these cases? Does the differential regulation through differentially regulate PSY, or there are some other mechanisms? So to answer this question, and we compared the OR hits and OR interaction with the PSY in yeast 2 hive. As you can see, both OR hits and OR Y type protein interact with PSY, and both of them also cause increased PSY protein level in callus culture. And we also looked, again in collaboration with Ralph Welsh from Germany. He looked at the PSY enzyme activity from OR his line with OR, and we found there's no difference. So which indicates that the differentially induced carotenoid accumulation is not due to the OR his has a high capacity to increase the PSY activity or increase the biosynthetic activities. So previously in our study, we showed that with the OR mutation in cauliflower, with the single gene mutation, it caused a change from numerous proplastic into 1 or 2 large chromoplast in the curd influenced meristem cell and also in the shoots, epidermis shoot cells. Similarly, our collaborators from Israel, and they also observed in mellow case, if you look under light microscope, and there's 1 or 2 large chromoplasts per cell. Like if you look at the tomato, each cell contains a huge number of chromoplasts. So both of them showed that the mutation or the later variation can induce large chromoplast formations. So what happened with Arabidopsis cali that showed the difference, big difference with carotenoid accumulation. And we look at this is lung transgenic Arabidopsis cali, and you can see there is a plastic contains some dark circle that's plast globule. And with the overexpressed OR gene, the plastic become larger, but they retain the same shape, but the plast globule become darker, and with the high electron dense. this is the same as PSY only expressed. We believe carotenoids are accumulated there. But when you look at the ORS mutation or other ORS mutation, and you can see the plastic shape changed from those and become the membrane structures. Actually, those membrane structures, membrane chromoplasts show exactly the same as what we observed in orange-curd cauliflower. So those experiments showed that the key for the specific high level of carotenoid accumulation is due to the unique ability of the ORS in induced chromoplast biogenesis. Actually, this experiment is fairly significant in another way. We know Arabidopsis are excellent model system to understand genes signaling in many processes. However, Arabidopsis doesn't contain a tissue with chromoplast. So essentially, we show here now we generate Arabidopsis lines that has a program to be able to form chromoplast, therefore provide us a system to better study genes and signaling in chromoplast biogenesis. So here's from our research, we showed that the OR mutant has dual functions in induced carotenoid overproduction. One is as post-transcriptional regulators to regulate PSY enzyme activity, and a larger one is actually the most important is to have unique ability to induce chromoplast biogenesis. And because chromoplast provide a storage sink that can stably storage carotenoids, at the same time it also stimulates continual biosynthesis. That's why the lines those plants or those lines can have a high level of carotenoid accumulation. Currently, we still focus on OR because till now, OR still is the only genes that know to convert chromoplast biogenesis. So, but how this OR gene control chromoplast genesis is still unknown. So this is a topic and we are focused on now. So in addition to those research, we also take the advantage of various omic tools to discover genes and proteins required for high level of carotenoid accumulation. So one experiment we did is protein omic analysis of chromoplast proteins in fruits and vegetables. As you can see here, we extract chromoplast from some of carotenoid-enriched fruits and vegetables, and we are hoping to looking for the common mechanism involved in those carotenoid-enriched crops for chromoplast biogenesis. So we separate the chromoplast protein from those vegetables and fruits. And one thing interesting, we found that if you look at the all chromoplast, the ATP synthase and the ATP translocators are most abundant proteins in those in all of those 6 chromoplasts. So the high abundance of ATP synthase and the nucleotide translocators suggest an important role of them in control or energy production and transport in control chromoplast biogenesis. And the one project is going on in the lab is we trying to figure out if the ATP and the ADP translocator is indeed involved with chromoplast biogenesis. In addition, we also did quite a lot of RNA-seq experiment in collaboration with many colleagues on campus. And one of the experiment is the bulk segregate RNA-seq transcriptome analyze. So again, in collaborate with Israeli scientists, we use the same lines to study the OR gene in melon fruits. So what they did is they grew the homozygous F3 family plants, and they extract the RNA from 25 orange or green fruits at various fruits developmental stage, and actually here are the RNA samples showed there. So we bulked those RNA and did the transcriptome analyze. And here showed the carotenoid metabolic pathway gene expression pattern. And we know that the mellow, the orange mellow accumulate a high level of beta carotene. We found nearly all the upstream genes are upregulated during fruit development, similar as the fruit mature. And the downstream genes are downregulated during fruit development. But very interestingly is in orange fruits and the carotenoid accumulate. But for the green fruit, there's no huge carotenoid accumulation, but the gene expression pattern is similar, which indicate additional mechanism. In this case, we believe the storage sink mechanism difference between the green and the orange fruit to distinguish the two kinds of fruit varieties. And in addition, in collaboration with Zhongjun Fei at BPI, he helped us to analyze all of those transcriptome data. And in addition, he also did the sleep analysis of the orange and green bulks. And with this bulks, essentially we only got very low, only 64 slips that's with over 90% of rate to distinguish orange and green bulks. And within those SNP, only two genes. One is the OR gene, and there is another one. It's totally, completely distinguished, 100% distinguished between the bulks. So if we don't know the OR gene is responsible for the orange color through the bulk segregate RNA-seq analysis, we would identify the OR gene, which also showed effectiveness of the bulk strategy for gene discovery. And another co-segregated gene, currently a visitor scientist, Hongmei Luo in the lab, is studying that gene to see if it also contributes to the carotenoid accumulation. So we looked at the differential expressed gene at different fruit development stage, and we found that there's only one gene showed differential expressed in all fruit development stage. But we are mainly focused on the 20DA2, and we select a few candidate genes, candidate genes for functional analysis. As I told you, the Arabidopsis colors system we used to start the OR function, and it showed very responsive and effective to response the genes that's involved with carotenoid accumulation. And we all know that the Arabidopsis with flowering dipping method, you can simultaneously transform a lot of genes. So this system also really give us a relative high throughput to test those genes and the proteins identified from our omics. And in addition, as I told you, that in many cases it's no OR control the orange versus non-orange fruit flash color. But within the OR germplasm collection, there are several several magnitude variation of beta carotene levels. So again, in collaboration with the Israeli scientists, we are going to genome type those germplasm collection by GBS, and then look the association between the mellow fruit beta-carotene content and the later genetic variation, not only to identify some markers to be used for mellow breeding, but we are hoping to identify additional alleles that control a high level of carotenoid accumulation. So in summary, our data shows that the OR mutant can serve as an effective genetic tool for carotenoid enhancement in food crops, both through traditional breeding and both through genetic and traditional breeding approach. And also OR mediates beta-carotene overproduction through regulates both PSY with enhanced biosynthesis ability and also Chromoplast with storage abilities. And as I mentioned, we believe that if we can put the carotenoid into Chromoplast instead of amyloplast, we can it can significantly help to stabilize the beta-carotene in staple crops. And another thing, our data shows that post-transcriptional regulate of PSY represent a larval mechanism under carotenoid accumulation. So finally, I'd like to thank co-workers. Here's the people currently in the lab. And especially I want to thank Hui Yuan, a postdoc in the lab, for the work I described, and also some former lab member for the work I described here, especially Xiangjun Zhou, and also an undergrad student, Katie Ostring, and she did 4-year research in the lab. She's a co-author of 3 of our publications. And she was a graduate last year with the highest honor, and her honor thesis research I mentioned briefly, and now she's a MD, PhD student in a very prestigious PhD, MD program. Finally, I also like to thank some of my wonderful collaborators, those Israeli scientists and their students. And Ralph for mellow work, Ralph Welch from Germany for OR PSY work, and some Chang Xu and Xiuxin Deng from China for the plastic work, and also some long-term collaborators on campus for their contribution for the work I just mentioned here. And the funding from our research from our USDA-ARS base fund background and the USDA-LIFA grant. And thank you for your attention. I'll be happy to answer any questions.
This has been a production of Cornell University, on the web at cornell.edu.